are going to build on topics that we've talked about in the previous chapters. So we're still in the cardiovascular system world. So when we're talking about these blood vessels in the cardiovascular system, this is going to be a closed transport system. So remember when we first started talking about the circulatory system, I used the analogy of transportation from point A to point B, where the cargo is the oxygen, the blood cells are going to be the car, and the highway or the pathway that everything's going to be on is going to be the blood vessels. So when we're talking about these blood vessels, it is closed. So there isn't an opening. It's going to remain inside the closed system. And what happens with these blood vessels is when the heart contracts, that will provide the force or the gas to move the blood from point A to point B. And as the heart contracts, it will actually force the blood into arteries and through all of the vessels in your body. So that heart will provide the force to be able to do it. And as it's forcing blood into the heart through the arteries, through the vessels, these are going to be leaving the heart. So that's the place that we are going to start with this exercise. We are talking about the systemic circulation that we referenced and described last week. So this systemic circulation, when it leaves the heart, remember it is being pumped around the body and then it will be brought back to the heart. When it's being pumped around the body, we're going to start with this side, with what happens when it goes from the heart to the body, and then we're going to go to this side of the systemic circulation. So what happens when it leaves the heart? Well, it follows a general trend. After the blood leaves your heart, it will go through what are called large arteries. And this is going to be large plumbing. So if you think about underneath your bathroom, underneath your sink, these are large plumbing, large tubes. These large tubes will then connect with and branch to smaller tubes or small arteries. It's a closed system. We're going to go from large diameter to small diameter. Then these small arteries are going to go to arterioles. And arterioles are very, very small vessels. These arterioles will get even smaller and they will end up as capillaries. And when the blood vessel is called a capillary, this will be in the tissue. Capillaries is where we will have exchange from oxygen to carbon dioxide. So this is going to be working at the level of Krebs cycle or metabolism. Capillaries is going to be the smallest vessels in your heart. And these capillaries will dump into vessels that are called venules. Small, narrow tube. The venules will dump into a slightly larger tube called a vein. And these veins are going to dump into what are called the great veins. And then this great vein will dump into the heart. So to keep it easy, a lot of tricks of the trade are A is going to be away from the heart. So if it starts with an A, it is going to be the name for the vessel that is away from the heart. Whereas if it has the V, it's going to be toward the heart. But it's easy to think about the A. A does not mean oxygenated. A typically carries oxygenated, but not always. We'll talk about that one. We'll break it down in a minute. But A will carry blood away from the heart. That is a very true statement. So when we're looking at the types of vessels, vessels are a generic term for the item that carries blood. So the vessels, there are two types. There are going to be the arteries and the veins. So both of these are considered vessels. And these are going to be transportation of gases. So they will carry the oxygen all the way around the body. So an artery will carry oxygen, whereas a vein will carry CO2. So back up in here, an artery is carrying the chemical or the compound oxygen. And then when it goes through this process down here, your carbon dioxide will then be what it needs to be carried through the blood until it leaves through the lungs. So these both carry transportation of gases. Arteries will carry oxygen and veins will carry O2. Within our veins, within our vessels, we have two types. We have arteries and veins, but there is one type of artery that's going to be a capillary. A capillary is where the actual exchange of gas will happen. So this is where it will go from O2 to CO2 in the system. You'll notice an artery does not exchange gas and a vein does not go through the gas exchange. Only a capillary does gas exchange, whereas arteries and veins carry the blood and transport the body. So if we're going from all the way from the head, here is our heart, kind of looks like that. The blood is getting pumped out. He goes down to the tips of our toes. As he's down in the tips of our toes, this is our artery. Then he'll go through a slightly narrower. Here's our arteriole. And then he'll go through a teeny tiny, very skinny tube. And this is going to be the capillary. And when we get to the structure of the capillary, we'll talk about these guys. But this capillary will separate all the way inside. This is our blood. He's coming in this way. And this blood will go out through our capillary. And then when this blood is coming out, we will then have other gases come in at the level of our capillary. So to kind of blow that guy up, because he's pretty small, we're going to look at one of those walls. So if this is the wall with our blood, and it is coming through the system like that, I'm just going to go ahead and give another wall. There we go. If the blood is coming through this side of the capillary wall will be some sort of tissue. So in this example, it's going to be a tissue at the tips of our toes. And as the blood is coming through the system, it will leak out into our tissue. And what will leak out is going to be the oxygen and any nutrients that need to feed that tissue. And this particular point, it will go from O2 to CO2 and then into the blood, and then out through the rest of the capillary, you will have CO2 and waste enter the system. So when we're talking about gas exchange, we are talking about the level of the tissue, which is going to be at a capillary, not at an artery or a vein. So this is at the level of tissue. So next we're going to look at the microscopic anatomy of both an artery and a vein. So they are very similar. They have the same general characteristics, but they are slightly different because they have different kinds of functions. So in general, we're going to talk about the microscopic anatomy of a vessel because this covers an artery and a vein. The microscopic anatomy of a vessel is going to be 3 layers. And these are going to be essentially 3 coats to the actual vessel. The term that is actually used is going to be tunic. So there are 3 tunics. So again, 3 tunics is literally just 3 layers or 3 coats. And they're all arteries and veins will follow the same pattern. So in this analogy up here, we talked about blood coming through our system like this, where we're talking about our vessels as a tube. Well, if they are a tube, we're going to slice it right down this way, and we're going to look at our tube as a cross section. Let me blow it up and make it bigger. And if we're looking at it as a cross section, it is going to look like that. So this is our tube or our cross section. So this tube will have 3 layers. The most inside layer, the most inside internal layer is going to be called the tunica intima, the intimate layer. You also may see it as tunica interna, depending on the edition of your book. But the inside layer is called the tunica intima. It is going to be made up of a single layer of endothelium. So thinking all the way back to first semester when we talked about what types of tissues, this is a single layer of endothelium. The technical type of cell it will be is going to be squamous cell. So squamous cell was the really flat-like discs, one single layer of squamous cell. This squamous cell allows for rapid diffusion. So these are going to be close fit cells. And they are going to be a very smooth surface. So this surface right here is going to be smooth, not bumpy. And the reason it's going to be smooth is it is actually going to decrease resistance. So this whole thing right here, we're going to draw on this guy. Here is our inside tube. So this right here will be called the lumen. So remember, this is a tube. So the inside lumen is going to be the hollow layer, the empty layer. So this is just empty space right here. This is where the blood will actually flow through. We're talking about these tunics are going to be between the lumen and the outside wall. So this first layer, let's do a color, right here. The first layer all the way around. Ooh. This is going to be our tunica intima. Our inside layer. This layer is the layer that is actually going to be closest to the blood. So as blood is pushing through this layer, the blood needs to have a very smooth surface to flow against, and it will decrease the resistance so the blood flows more smoothly. That's the purpose of the tunica intima. Okay, the next layer is going to be the tunica media. And it's just like the name. It is the middle layer. And the tunica middle is the middle layer. And it is going to be made up of 2 main types of tissue. It will be made up of smooth muscle, and it will be made up of elastin fibers. He is going to be connected to the sympathetic nervous system. So this layer is made up of smooth muscle and elastin, which means it has the ability to stretch and recoil. And it's connected to the sympathetic nervous system, which will give it instructions on that smooth muscle contracting or that smooth muscle recoiling. And the reason that this is super important, the smooth muscle and the elastin and the connection to the sympathetic nervous system, is because this will regulate the diameter of the lumen. So if we have a blood vessel that looks like this, and we have a lumen that looks like this, essentially this right here would be all smooth muscle. But let's say we want to make our diameter larger. So here's our lumen, our cell. We can do this to our lumen, and we can make essentially that smooth muscle that's in this middle layer contract. And it will make the walls of your lumen pull out. So this is where it was before, and this is where it is now. So when we increase the diameter of our lumen, more blood can run through. If more blood can run through, we can regulate our heart rate. So coming back up to this guy, if that's our tunica intima, change colors. Our tunica media is going to be a large, bulky layer made up of smooth muscle and elastin fibers that makes up this layer right here. And if he contracts, he can change the size of the lumen. The last layer is going to be the tunica externa, sometimes also called adventia. And this is the outermost layer. So this is the external layer, i.e. tunica externa. Tunica means layer. He is going to be composed of another type of tissue. He's specifically going to be made up of areolar and fibrous connective tissue. So these two combined give it the ability to be a little bit more of a harder surface. Not connection. Ooh, so sorry. Is there an undo? Yes, there is. Connective tissue. So these two combined allow it to have a little bit more of a denser surface so it won't collapse. So the outside wall has the ability to be supportive, and that is the function of this guy. He is support, he maintains the integrity of the wall, and he also protects because he is a thicker layer, and so he'll protect the outside. So coming up here, our tunica externa will be a layer that looks like this all the way around your blood vessels. So these are going to be the general 3 layers that all of your vessels will have. So this includes your arteries and your veins. But there's a couple differences between your arteries and your veins. So I'm just going to call them A plus V. They follow that same pattern, but they have a couple differences to them. Some differences are going to be your arteries are thicker than veins. So we're going to do all your artery stuff over here. And the reason your arteries are thicker than your veins is because the tunica media, that middle layer, will contain more smooth muscle. So it's going to be thicker because the middle layer is bigger. Your veins are going to have thinner walls. Just in general, the veins are going to have thinner walls. And the reason they're going to have thinner walls is because these guys are further away from the heart. They do not need to have that really thick, smooth muscle layer because they do not need to pump blood all the way around the body. With these veins, you will also have a larger lumen, so a larger space. And one other thing that's interesting about veins is because they are going back to the heart, they are usually going against gravity. So a vein will actually have valves in them. And these valves will prevent backflow. So the blood can't dribble back down. It's caught by a valve. So when you're looking at these underneath the microscope, and I believe there's a drawing for this week, this is going to be kind of what an artery looks like, whereas a vein may actually look like a collapsed piece with a little bit of a smooth muscle all the way around it. But larger lumen, smaller lumen, increased smooth muscle, decreased smooth muscle. And this is usually going to be circular, nice and round, and this will normally be collapsed. So pulse lives in the world of the cardiovascular system, and pulse is the alternating pulse of pressure that is going to be against an arterial wall. So if we are going to have all of the blood coming through our heart, leaving our heart, here we go. Our blood is leaving our heart. It's now rushing through our vessels. As it's going it's going to hit the walls of our heart. The walls of our blood vessels as it goes. That is what we are measuring through pulse and blood pressure. So to break it down a little bit, when we are actually looking at, when we're looking at pulse, how do we actually measure this, guy? So pulse is going to be done off of systolic pressure over diastolic pressure, which means the pressure when the blood is leaving the heart because the heart is contracted. Remember, that's what systole means. And it's going to be the heart contracted versus diastole, which is going to be the heart relaxed. So technically with pulse, you're measuring the pressure when the heart is contracted versus the pressure when the heart is relaxed. So that's what you are doing in that world. The average pulse of an adult is going to be between 70 to 76 beats per minute. That is what you will hear with this heartbeat. The pulse should relate to that. When you're looking at pulse, if we have a little person who looks like this, and we're going to make him happy today, a couple places that you can detect pulse really, really well. You have a radial pulse. And a radial pulse is going to be where your radial bone is, a radius. So that means it is typically going to be taken at the end of the radius. The main place for the radial pulse when you're taking this one is going to be between the arm and the hand. So this is the wrist. When you're taking the wrist pulse, you're taking the radial pulse. Another place you can take the pulse that is going to be the brachial. This is also going to be similar to the name of the bone. So that's going to be right here. This is technically going to be at your elbow. When you take the pulse right in the crook of your elbow, you're going to be doing it at you're measuring your brachial pulse. And then you're also going to have a common carotid pulse. When you are taking the common carotid pulse, you are actually taking it right here. So this is the one that is going to be in your neck. So when you go and work out, or really when I was in elementary school, that's when I remember doing it the most, and you put the two fingers on your neck, what you're actually measuring is the pulse of your common carotid artery, so your common carotid pulse. All of these pulses can flux slightly because if your heart is in the center of your chest here, depending on whether or not you are measuring pulse this close, which is very, very close, your common carotid is very close to your heart, whereas your radial pulse is farther away from your heart, there can be a little bit of flux. It is minute in the grand scheme of things, but you can detect a difference sometimes in the pulse with where you take it on the body. So another So when we are looking at this measure of the blood pressure against our artery wall, we're doing our systolic pressure over our systolic or diastolic pressure, and we're looking at our beats per minute with our pulse. We're also going to be measuring these pieces in milligrams per mercury. So we're going to take our blood pressure now. And I apologize, I skipped way ahead of my notes. So essentially, this whole little section right here, I skipped way too far ahead. This is going to come down here and be our blood pressure. This is talking about our pulse. Pulse is 70 to 76 beats per minute. You can take them at a couple different places. Those are three major ones. When we're going to do our blood pressure, I apologize, that is where we are going to be doing our systolic versus our diastolic. So when we're measuring this is normally going to be measured in millimeters per mercury. Millimeters per mercury. This is an older way of measuring. It's very similar to how temperature used to be taken with the little glass thermometers that you would hold under your tongue, millimeters of mercury, how much the mercury would move in that little glass vial. We no longer measure it this way with the actual tool, but it's still done in millimeters per mercury. So that's what that mmHg means. But we are measuring the systolic pressure of the heart when it's contracted divided by the diastolic pressure when the heart is relaxed. So this is usually going to be around 120 over 80. So 120 millimeters per mercury over 80. This is considered normal. I've heard recently that they're actually recommending 110 over 70. I'm not that involved in the current clinical world, but 120 over 80 or 110 over 70 are both considered normal for blood pressure related concerns.